Lab 5 Analysis of Breathing Data by Meagan Sullender and Clift.
Respiratory system. The respiratory system is responsible for bringing oxygen to the blood and removing carbon dioxide. This is done by a vast system of capillaries that run throughout the alveoli, which are air pockets inside the lungs. By enlarging the surface area of the lungs, the alveoli make for fast and efficient gas exchange. Inside the lungs, a surfactant is produced in order to relieve surface tension of the liquid layer on the alveoli and keep the lungs from collapsing with every breath.
The blood and respiratory system work together to maintain a balanced pH in the body. This is done through the carbonic acid bicarbonate buffer system in the blood. A buffer is a substance that tends to resist change in pH when given a quantity of H+ or OH-. The buffer system is made of conjugate acids and bases and forms an equilibrium system. This system holds the pH of the blood at a constant value. If the pH of the blood falls below 7 or above 7.8, irreparable damage occurs, such as loss of cell structure and function, as well as to the catalytic activities of enzymes.
In the bicarbonate buffer system used, the proton donor species is carbonic acid, is in reversible equilibrium with the dissolved CO2, as shown in the equation below. This equilibrium is regulated by breathing. Slow or shallow breathing increases carbonic acid concentration, while fast or deep breathing decreases the concentration.
The objectives of this lab are to understand the acid-base balance in the body by comparing CO2 concentrations of the lab air and exhaled air with both lime water and phenolphthalein, examine lung capacity through the use of a spirometer, and to demonstrate a knowledge of statistics.
For this lab, we hypothesized that lime water will get cloudy with exhaled air and remain clear with lab air. A color change would occur for all three trials, lab, rest, and exercise, but would be the fastest for exercise and slowest for lab air. No color change with lab air, no color change with exhaled, and with a more significant color change after exercise. Vital capacities will be close to average for age and height because neither member had breathing disabilities.
We performed protocol listed in BB 2903 Lab 5 Dissection 3 procedure. The only exception was in Part B. 30 milliliters of phenolphthalein solution was used in place of 200 milliliters per beaker.
For Part A, 2 beakers were filled with 50 milliliters of lime water were prepared. Air from the lab was bubbled through the beaker for 1 minute using a pipette pump. Exhaled air was bubbled through the remaining beaker for 1 minute using a straw. Observations were recorded in the table below. Results show that the beaker bubbled with room air remained clear after 1 minute of bubbling. The beaker bubbled with exhaled air became significantly cloudy after 1 minute, with cloudiness starting to appear after 40 seconds.
These results support our hypothesis. When exhaled air, CO2, reacts with lime water, CaOH2, calcium carbonate, CaCO3, is produced, which makes the water cloudy. Since the amount of CO2 in the air is minuscule compared to the amount in exhaled breath, very little calcium carbonate was produced. This is why the lab air beaker stayed clear, while the exhaled air beaker became cloudy.
For Part B, 3 beakers were filled with 30 milliliters of phenolphthalein each. The lab air was bubbled through 1 beaker until the solution changed from pink to clear. Rest air was bubbled through a 2nd beaker until solution changed from pink to clear. After 2 minutes of exercise, air was bubbled through the 3rd beaker until solution changed from pink to clear. Once each solution was clear, drops of ammonia were added until the solution turned back to pink. The amount of ammonia needed for a color change in each solution is directly proportional to the amount of CO2 in said solution. Table 2 shows normalized number of drops of ammonia used in each beaker in order to turn the solution back to pink. The equation for normalizing data is the time in seconds for room air to turn clear divided by 30 seconds, all multiplied by the number of drops of ammonia for exhaled air.
Table 3 shows the average values for the normalized number of drops of ammonia needed to turn the solution back to pink for each trial, as well as the standard deviation. While our individual values are not necessarily the same as the class averages, they fall well within the standard deviation. The standard deviation for each trial is so high due to the fact that a new carboy of phenolphthalein was not made for each class section. The longer the carboy sat in the room, the more CO2 it absorbed from the atmosphere, changing its color and causing variability in data.
The data in both data table 2 and 3 support our initial hypotheses that the solution would turn clear faster with the exercise air and slower with the lab air in relation to the rest air. This was because the The most amount of CO2 was exhaled after exercise. A significant amount of CO2 was exhaled while at rest, and a very minute amount of CO2 is present in the lab atmosphere.
For Part C of this experiment, each group member inhaled to capacity and exhaled into the spirometer in order to measure the vital capacity of the lungs. This was repeated for 3 trials for each group member. The recorded measurements can be found in Table 4, as well as the average taken from all 3 trials, along with the general average of all the public for each member's height, age, and gender. Since the average for all the trials for each member was approximately the same as the estimated value for our height, age, and gender, our hypothesis was supported by the data.
These are our references cited throughout the lab.
Thank you.